Hi everyone, welcome to my channel Pure Biology, and today I come with another video that is the insulin signaling pathway. Okay, so it's a topic related to obviously cell signaling process, and here I will try to explain this process very briefly. So continue with my channel, okay, till the end of the video. Okay, so here the insulin receptor, this is called the INSR. This insulin receptor is a receptor tyrosine kinase, okay, and it consists of two alpha subunits on the outer surface and two transmembrane beta subunits. So it consists of two alpha subunits and two transmembrane beta subunits. The alpha subunits that present in the outer surface or outer face. The two alpha subunits that are linked by the disulfide bonds, okay, and there is single disulfide bond between alpha and beta subunits. That means disulfide bonds that present between two alpha subunits and obviously alpha and beta subunits. Clear? So the two alpha subunits, they are joined together by the disulfide bond, and alpha and beta, these subunits also joined by the disulfide bonds. The binding of insulin to the alpha subunits, that means when insulin binds to the alpha subunits, that triggers a conformational change that allows autophosphorylation of the tyrosine residue. Okay, so what happening, that when the insulin binds with the alpha subunits, that time a conformational change is occurring and also autophosphorylation starts. Autophosphorylation of the tyrosine residues that starts. Clear? And in the carboxyl tyrosine domain, So what I am saying, that the insulin that binds with the alpha subunits, and at that time the autophosphorylation of the tyrosine starts, and in the carboxyl tyrosine domain of the beta subunits. So when the alpha subunit and insulin binds to each other, at that time autophosphorylations of the tyrosines that occurring, that tyrosines are present where? They are present in the carboxyl tyrosine domain of the beta subunits. Clear? And the phosphorylated insulin receptor next phosphorylates insulin receptor substrates. Okay, you just ignore the sounds because it's raining in the outside. Okay, so here the phosphorylated insulin receptor next phosphorylates to that insulin receptor substrates. Okay, insulin receptor substrates, and that are present on the tyrosine residues. Clear? And the phosphorylated IRS or the insulin receptor substrates. Okay, so this tyrosine phosphorylated IRS that binds with the SH2. When the IRS that becomes phosphorylated, they binds with the SH2 domain. Okay, and this SH2 domain that present in the proteins, and that are directly involved in the mediating different effects of the insulin. So you just mind it that this IRS that binds to the SH2 domain of the proteins. Clear? And this phosphorylated IRS that acts as docking site for enzyme PI3 kinase. Okay, that means the PI3 kinase first phosphorylates and to form phosphorylates PI3 bisphosphate. So whom this enzyme PI3 that phosphorylates this PI3 bisphosphate, or you can say PIP2, and it converts the PI3 bisphosphate, Pi3,4 bisphosphate into Pi3,4,5 triphosphate or PiP3. And this PiP3 when binds to the PKB, that leads to the activation of the PKB, or that PKB that gets activated when PiP3 binds with this PKB. And this activated PKB phosphorylates and to the glycogen synthesis. Okay, so when PKB is activated, this phosphorylates the glycogen synthesis kinase 3, that is the GSK3. Okay, and inactivate it. Okay, so when PKB that phosphorylates GSK3, it's become inactivated, and now inactivated GSK3 that makes the glycogen synthesis active. So when GSK3 become inactivated, then the glycogen synthesis that becomes activated, and due to the activation of the glycogen synthesis that accelerates the synthesis of the glycogen from glucose. Okay, so when GSK3 that becomes inactivated, in that situation glycogen synthesis that becomes activated, and when glycogen synthesis becomes activated, this accelerates the glycogen synthesis from glucose. So now glycogen is stored within the liver. Okay, and in case of muscle and fat tissue, then PKB also triggers the movement of the glucose transporters. These are called the GLUT4 glucose transporters, and from the internal vesicle to plasma membrane and stimulating the glucose uptake. So that means here in the muscle and fat tissue, this PKB that triggers the movement of the glucose transporters and that stimulating the glucose uptake. Okay, so this is the process of the insulin signaling pathway, and I just briefly explain this topic. So you just consult this, first see this video, and after you can consult any cell biology book for getting extra knowledges about this topic. Okay, so thank you, and continue with my channel.